Introduction of operation method of spectroelectrochemical measurement, using SEC-2020 Spectrometer system and Model 2325 bipotentiostat. Assembly of spectroelectrochemical cell. For the spectroelectrochemical cells, there are batch type, SECCT, and flow type, SEC-3F. In this video, we will introduce how to assemble the SEC-3F Spectroelectrochemical flow cell. The assembly image of the SEC-3F Spectroelectrochemical flow cell is shown. Here are the details of the components. Flow cell block 1. Flow cell block 2. Silicon gasket. Pan small screw. Teflon tube. Stainless steel pipe. Fitting peak. Silicon tube. And needle for suction. Working electrode and reference electrode are options. Phillips screwdriver is necessary for the assembly. Overlap a gasket on the flow cell block 1, matching the screw holes. Set a working electrode to cover the center hole of the gasket. For ITO electrodes, face the front side, electrode surface, toward flow cell block 1. Let it protrudes about 3 mm from flow cell block 1. Placing another silicone gasket over the working electrode. This will prevent liquid leakage from the cell. Set flow cell block 2 over the flow cell block 1 and the gaskets, matching the screw holes. Fix the flow cell blocks 1 and 2 by 4 screws, while making sure that the working electrode does not slip off. Tighten the 4 screws evenly. If you tighten the screws all at once, the working electrode may crack, so tighten them little by little. Insert the stainless steel pipe into the fitting. Put out the tip about 2 mm. Connect the silicone tube to the stainless steel pipe. Cut the Teflon tube to a suitable length and connect it to the silicone tube. Insert another Teflon tube into the fitting. Stick out the tip about 2 mm. Attach the Teflon tube for waste liquid to the X position in the figure on the upper right of the screen. When attaching the fitting to the flow cell, be careful not to over-tighten the screws. Install the stainless steel pipe at the Y position in the figure on the upper right of the screen. Attach a reference electrode at the Z position in the figure on the upper right of the screen. The stainless steel pipe is also used as a counter electrode. If the solution is sent from the inlet side, bubbles are less likely to form. If you do not use a liquid feed pump, please use a suction needle instead of a Teflon tube for waste liquid. Attach a fiber collimator included in SEC-2020 on flow cell block 2 side. Remove the inner cylinder of a collimator included in SEC-2020. Attach the collimator to flow cell block 1 side. Next, set the SEC-3F flow cell on SEC-2020 platform. Let the fiber collimator facing the spectrometer. Connect the fiber collimator and spectrometer. Insert the light source into the collimator with the inner cylinder removed. Finally, fix the position of the light source and spectrometer with the screws for the platform. This completes the assembly of SEC-3F Spectroelectrochemical flow cell.